Lo único que puedo decir es que es una experta en biomateriales para biomedicina, tiene una trayectoria profesional muy larga, hizo la tesis en la Universidad Complutense, después estuvo, si no me he enterado bien, en Northwestern University, en el Instituto Carlos III, en el Hospital Nacional de Parapléjicos, y ahora tenemos la grandísima suerte de que sea una compañera nuestra en el Instituto de Ciencia de Materiales. O sea que... ¿repito todo? No. Bueno, pues...
Nada, vale.
y hoy nos va a hablar sobre este tema que está aquí expuesto, que dice que...
Vale, muchas gracias, Luis. Thank you very much, and I take the opportunity to wish you very happy New Year. It's the beginning of 2026. I hope you enjoyed a very nice vacation break, and we are back. Now we have to get back to our work and our duties and so on. Today I'm very happy to be here to present you a Piezo4Spine that is a dream, still a dream. It hasn't been a reality yet, so spinal cord injury patients continue to be injured and there is still no cure for them. But for me it's been a dream, and also for many people that have been working with me for these years. As you will see in my presentation, I will expose some of the results that we are getting, but also some of the trajectory that gave me the pathway to really get to here. I'm the coordinator of this project. It's been a challenge, but it's also a pleasure to be there and to be learning together with my mates. Piezo4Spine is a Pathfinder project. We are already in the beginning of the 4th year, so we still have about 12 months to complete as much as we can. It's always, as you know, impossible to achieve all the objectives that one writes in the proposal, right? Because we want to be super happy about the excellent science that we will be doing, but the reality gives back, puts up us back into the earth, right? And then the scientific objectives sometimes result very difficult, but we are advancing a lot and we are learning very much. As you can see here, we are 7 different institutions. CSIC is coordinating. We have a very important, the National Hospital for Paraplegics. You will see that later. That is a key piece of our project. We have universities, the University of Louvain and the University of Coimbra. And then we also have the Italian Institute of Technology and 2 companies. One that is developing 3D bioprinters that is in the field that we work on, and then also this Austrian company that has been very helpful for all the communication activities in the project and is having a very important role in there. I also like to note this aspect about the participation of women, because it's also a very important challenge for us to get women in the leading positions in the projects. So I achieved that, and actually 4 of the 7 teams are led by young women that are also demonstrating their value. So these are the faces behind the logos that I just showed you. As you can see, this is definitively one of the strongest pieces of our project. That is the strong consortium that we have. It's multidisciplinary, interdisciplinary, and complementary. You see here some faces that you know, Puerto, Ricardo, and myself, but they are also very important people. Beside us, there are Fabio Viscarini from the Italian Institute of Technology, that is an expert in organic electronics. We have Juan Aguilar and Juliana Martins, that are neuroscientists working in the National Hospital for Paraplegics and experts also in neuroscience in general and spinal cord injury in particular. We have our clinician here. It's fundamental for any research in materials for health that we really understand the problem that we try to solve, in our particular case, a spinal cord injury, and we have here the expert, the one that really knows about the spinal cord injury. If you think that I may know something about a spinal cord injury after my talk, you don't have an idea of what she knows about that, because she treats those patients every single day for the last 30 years. So she really knows what a spinal cord injury is. Then we have Alino Ferreira from the University of Coimbra. This is an expert in advanced therapies and biotechnology in general. And Andreas Blesser, that is the CTO of the company, the German company that is building these 3D printers. We have one in our lab, in the cell culture lab, and is developing bioinks to help the project. Here are some very nice pictures that we took during the annual meetings, you know, that we have to do in these projects. This is back in 2023 when we met in Ferrara, then in Coimbra in 2024, last year in October in Brussels. But the outline of my talk is not really the science, it's the people, the young people that have been working with me during these years and that really made possible that I'm here today and that I progress all that I have done. So you see here the name of the people that built all the results that I will be presenting. Ancor González, Formación Profesional Technician, that worked with me from the very beginning in this topic back in 2014 when I left this institute as San Juan de la Cierva to join the hospital as San Miguel Servet. He was hired by my project and we built together most of the story that I will be presenting today. Ana Dominguez, that was my first PhD student that was in the lab for 4 years. André Girau, that came from Portugal to join in a combined PhD program and was also doing a very fantastic work in the lab. And Arche, co-shared with IMDEA Nanociencia, also a fantastic worker, contributing very much to a lot of work. Esther Benayes, that is still in the lab, and we hope we have a fantastic graduation soon in this month. And Julia Martinez, that is still also in the lab and is expecting for a nice graduation also in 2026. She and all of us. Okay, so now we get to the target, spinal cord injury patients. Okay, spinal cord, you know, it's in our body, inside our backbone. Very important take-home message today. It's not a cable. I repeat this in all my talks because it's not a cable, and that's the reason why spinal cord injury remains without a cure. That's the reason why it's so difficult to find a cure for this, because it's an extension of our brain. So it's the central nervous system. All the complexity, all the high differentiation of the components, the very low regenerative capacity that it has is shared with the brain. And that's the reason why we still don't understand very much how it works. And it's so difficult to find a cure or any therapeutics that can really go there and solve the problem. So, unfortunately, you see here the numbers. We have between 250,000 and 500,000 new people that are suffering spinal cord injury every year. And then the prevalence and incidence varies depending on the countries. In the US, they have some of the highest prevalences and incidences. What means a spinal cord injury? It means that we lose partial or totally the sensing, the motor functions, but also autonomic functions. So you know all the organs that we have inside, they have innervation from the spinal cord. And that's also damage. So our intestines, our bladder, that stop working if we have a spinal cord injury. Even breathing. Breathing is controlled from the cervical segments of the spinal cord. So it's a serious thing. It's not just we cannot move our legs or we cannot move our hands. It's much more than that. And as I mentioned before, we have here this very important person in my life and in my work in the last 10 years, that is Elisa López Dolado. When I had to leave the National Hospital for Paraplegics, I was very, of course, happy because I got a permanent position. I cannot deny that. But also sad in the sense that the National Hospital for Paraplegics gave me really a target, no? A focus in my research, a very important one. So I didn't want to lose the connection with them. What I thought about, what I figured out to do, create a joint research unit with them. And that's what we have been doing and building during these past 7 years. It was accepted by the CSIC in 2017, and we have been renewing it every 3 years and working very nice. With Elisa, that is now the leader of the group that was created by me when I was there as a postdoctoral fellow, we continue getting funding and publications in the topic. There are many groups in the world that are working in spinal cord injury, trying to find therapeutics for them. In here, I just want to briefly mention the top ones, or the ones that I, from my modest knowledge, consider are on the top. The first is Grégoire Courtine. Grégoire Courtine is indeed the director of this Center for Neuroprosthetics and Brain-Mind Institute in Switzerland. And together with Jocelyn Bloch, that is a neurosurgeon, so very close to the clinicians that really know what is happening there, they are building a very nice piece of knowledge and work on what spinal cord injury is and how to approach the therapeutics. Their approach is a little bit different to our approach in the sense that they really focus on epidural electrical stimulation. They have been working on the development of electrodes to deliver that electrical stimulation in the segments that are affected to kind of bypass the segment of the neural tissue that is affected. They have been publishing very nice pieces of work. I will not stop much on that, but just want to mention that all these papers that they have been publishing in the last 3-4 years come from this clinical trial that was initiated in 2014, so 11, almost 12 years ago. This means, we already know that science takes time, and takes time also to get to the patients in the case of materials for health, also for any other topic, right? But in the particular case of health, when you want to develop a therapeutics that want to reach the patients, that needs time. And they have been working very hard and with a lot of money on this for over 10 years. And now they are really getting the seeds of that. I just want to mention that they not only want to cure the spinal cord when it's damaged, but also want to understand what happens there. That will help to develop the therapeutics, of course. In that particular case, I want to mention this work that is quite recent, 2024, in which they identified a particular area in our brain, that is the lateral hypothalamus, in which there is a portion, a population of neurons that are glutamatergic that have been identified to be related to any recovery of the walking. In particular, I want to mention, they proved that in rats, but they also proved that in patients, in spinal cord injury humans. Another important name in the field, Monica Sousa. We were lucky enough to have her 2 years ago. She came to the Institute to give a talk. She has been granted with an ERC Advanced Grant and is doing a fantastic work on the investigation on this rare and curious mammal, that is the African spiny mouse, that is the only mammal that is able to repair the spinal cord after damage. There are other animals, not mammals, that have that capacity, not many, but a few of them, but the only mammal that can do that is this spiny mouse. She has been working very hard on identifying what is different in that mouse, what makes that different to other mammals or to the rest of the mouse. And she has identified a few pieces. But what is probably happening is that there is not only one piece. It's probably different elements that combine make that spiny mouse able to repair the spinal cord. And another important point that I want to mention is that spiny mouse is not only able to repair the spinal cord, it's able to repair many other tissue, any tissue that is damaged, and it does very rapidly. So it's not only the spinal cord that has something different. It's the whole body has specific areas of the metabolic pathways that are different and make the response of after an injury not that much of an inflammation and paralyzation of the regeneration, but a rapid resolution of the inflammation and then the recovery. Another important name is Ronaldo Ichiyama. He also came last year in June to give a talk. He was actually in the origin of all the work that Grégoire Courtine did. They were together working in California, and he was the one who developed the electrodes. But then life, is different. Characters of people are different. And Grégoire was luckier and had other pieces or elements in the pocket, and he became the famous one. But he was on the back of those electrodes together with Reggie Egerton, that is a top neuroscientist and has done a lot of fantastic contributions in understanding the spinal cord. He is focused on epidural electrical stimulation, as Courtine does, but he also focused on the understanding and not to exposing the spinal cord to electrical stimulation, what is a very important aspect. And he's combining that with motor rehabilitation, that is what spinal cord injury patients receive every single day after the injury, because it's known clinically that it helps the recovery of certain functions. So that said, we go back to Piezo4Spine, and from the scientific point of view, I already told you that the consortium is strong, but also the idea, the radical idea and vision that we proposed was strong, is still strong because there have no papers that have really ruined out our hypotheses, are mechanotransduction and fibroblast-mediated scarring. Those are like the pillars of our developments. Regarding mechanotransduction, I must introduce the piezo-mechanoreceptors. In here, because the audience is a lot about material science, I need to separate that piezo does not talk about piezoelectricity or piezoelectric materials. It talks about piezo-mechanoreceptors that are transmembranal proteins, as you can see here. This is the 3D structure of this receptor. You see it has 3 different pieces, and it gets into in the cell membrane of all of our cells, and is able to sense the mechanical force. So when the mechanical force gets to the cell membrane, it gets deformed, and then the piezo-mechanoreceptors, they are identified 2 of them, 1 and 2, with a different distribution depending on the tissue. I will not focus on that today. What happens is that this mechanical force make a conformational change in the channel, and then we have the entrance of calcium. It's a non-specific calcium channel, but calcium is a fantastic, probably most of you know, is a fantastic intercellular messenger. So it gives the cells the capacity to translate mechanical force, physics into biology, into messages, into the the roots of the cells to do things, to do biological responses. The other target, sorry about that. We think in belief, and still believe, that by targeting these mechanoreceptors, we can get a faster recovery. And then we have the fibroblast-mediated scarring. This is well known for decades. When the spinal cord injury gets damaged, we get a rapid formation of a scar that is positive in the sense that avoids that damage to get extended in our central nervous system, what is very important because we don't want to get the rapid death of the person, but at the same time, is negative because it hampers neural regeneration. Through the scar, the neural tissue cannot grow. The signals that it gets, the neurons, the astrocytes that are around, don't grow through here because it's damaged. And that's what we want to eliminate. We don't want that scar, or we want a scar that tells the neural system when it's stabilized, yes, you can go through. You can grow in the middle of this because it's no longer an insult. It's no longer a problem for us. All these ideas are also based on my background as a scientist, that is tissue engineering. From my very beginning as a PhD student, all my research focused on these 2 words that are tissue engineering. So this is a kind of an old concept already. I will turn 20 years old from my PhD this year, so I'm not that young anymore. My white hair can tell that. But this has been with me for my entire life. And this concept that came from the hand of these 2 pioneers and great men that are Professor Langer and Professor Vacanti was created late in the 90s as this new concept of combining disciplines. No? This super nice word that we like to use now, multidisciplinary, it was back at that time. They really combined that. And it was, as you can see here, combining principles of engineering and life sciences all together to make a substitute of an organ, of a tissue that is damaged. And it's damaged and cannot be cured because we cannot replace any organ in the body. There are organs that cannot be easily replaced, or they are not sufficient donors. So we need something else to replace. And that's where this idea came, and these scientists and all their teams really developed. But this has evolved as everything in life, right? And then we have these new figures, Professor Laudenstein and Professor Amir, who also came here in August to give a great talk on the concept of regenerative engineering and the great work that his lab is doing. The natural evolution of tissue engineering, in which we really converge a lot of different disciplines and all of that to get a clinical translation. That is what we really want. This field has been running for over 30 years, but there are still not that many things that has arrived to the clinical, no? In bone, in muscles, there are many of them, but in neural, for instance, only the peripheral tissue, and that's because of the complexity. So we really need to move forward. And another important element in the research that we have been doing in the last decade is graphene. I will not explain much because there are in the audience people that are, many people that are much more of an expert on graphene than I am, because I'm a biologist and actually I'm just using it as a tool, so I will not mention much other than what I really like is reduced graphene oxide. So I like the defects. We all are imperfect, and graphene, as it is perfect for many different applications, but for biology, I prefer it to be imperfect. I prefer to have oxygen groups that can be functionalized and can make the material a little bit more hydrophilic, because biology comes with water. So I like my material to be nice and happy in the presence of water. Said that, there is a lot of papers that already demonstrated how positive is the interaction of these graphene-based materials with the different with components of the central nervous system. This in vitro, this other in vivo. It does not interfere with the neurogenesis. It helps the migration of progenitors, as you can see here. So many positive findings, and that's the reason why back in 2014 I thought about, actually a little bit earlier than that, I was still here in the institute, thought about introducing this material in neural repair. There are important contributions on the use of graphene by other of our mates, in this particular case Jose Garrido from the ECN2. They published this very nice piece of work in 2024 in which they used this graphene for the development of microelectrodes, actually for in vivo high resolution neural recording and stimulation. And as you can see here, they get a very high fidelity recordings, and they are able to stimulate low current thresholds and high current thresholds and high selectivity, and giving that, achieving in a very small size of microelectrodes, as you can see here, 25 microns and low impedance. So, what we have done during these years, a natural evolution, as I mentioned before, for the concept of tissue engineering itself. We started just with the simplest, just putting a little bit of graphene oxide on top of a glass cover slip and to culture the neurons on top to see how that worked. The neurons were happy, so we were happy also, and then we continue progressing. We jump rapidly to the 3D. Why? Because we are trying to replace tissues, and tissues are 3D. I don't need something that is 2D. For other applications, it's nice. Yes, as small as possible. But in my case, I need something that is 3D, because the tissue developed in 3D. We also tried 3D microfibers. Why? Because many people were doing that. But we were not having very nice results. So we went back to really our 3D porous scaffolds. That will be the results that I will be presenting. So, and here comes the people, Anko and Anna, very good and hard workers, set up this model in which we were trying graphene. You see here, we replace half of the spinal cord at this level, the cervical level, and then putting here our scaffold. And then evaluated it at different time points. First, we started the shoulders, so subacute, only after 10 days, and got very nice results. Then more chronic, let's give it the rat 1 month to see what it happens. Also very positive results with immunomodulation and angiogenesis. And then to a longest time point, 4 months. At that particular time point, what we found that you can see here, this is a transversal cut of the spinal cord. You see here, this is an injured rat. In here, all this whitish color that you see there, that's a scar. The famous scar that I was talking about a little bit earlier, this is this. As you can see here, there is no possibility of neural growth at all. What we got, these are the rat, this is a transversal cut of the spinal cord of a rat that has received our reduced graphene scaffold. You see here, much less white color. There's still something, of course, but much less than here. And in here we have a lot of space that is our scaffold that can be colonized. So this was very positive, but it was even more positive to find that in other other areas that were not exactly at the lesion site, but a little bit away from the lesion site, we found that the number on the maximum area of the lesions that still are showing still damage, what we call perilesional damage, were much reduced in the rats that had our scaffolds in comparison just with the injury. So it's not only a positive effect at the lesion site, but if we move a little bit farther from that area, we also get a positive influence of our scaffold. We also found angiogenesis after 4 months. Our scaffold, you see here, this is a scaffold, a lot of green coloring here, that's RICA1, that is a marker of mature endothelial cells. Endothelial cells are the components of the lumen of the blood vessels. So we had functional blood vessels in there. You see here a red blood cell inside of some of the blood vessels that were growing inside our scaffold. We get vasculature inside our scaffold, we get blood, we can grow. The tissue can grow because it has blood supply, the removal of all the things that the waste materials that we don't want, but also the arrival of nutrients. And we also found neurites. As you can see here, this is again the scaffold, and you see a lot of reddish, whitish, and greenish color. That means different markers of neurites. So our scaffold was also colonized by neurites coming from the borders of the injury. And they were actually myelinated, as you can see here, because they were more mature for the transmission of the electrical signal. So another important point that we need to check at that time and that we continue doing is the toxicity. There were no signs of systemic toxicity in these rats after 4 months of implantation. We checked the main organs, you see here, and also some other organs as the heart and the intestines, and we couldn't find any damage. So this was the starting point for Esther and for her PhD work. And she challenged the system a little bit further. Instead of just removing half of the spinal cord at the cervical level, let's make a complete cut. Let's make it serious. A complete transection, as you can see here. So we cut a piece, and then in that segment, we implanted our material. It's similar material as before, with the electrical small, but some electrical conductivity, the same porosity, exactly the same material. And then we use, for the first time, electrophysiology to really see if we could get communication. In our previous model, we couldn't do because there was an unaffected or supposedly unaffected half of the spinal cord that could transfer the signal, right? In this case, no. We remove completely. So if there is any response in the brain after a caudal stimulation, it has to be because our scaffold is doing something. It's really connecting with the top part. And that's what we did. And you can see here the traces of 4 particular neurons that are located in the reticular formation that were responding to our stimulation caudal to the scaffold. So electrical stimulation through here, and then response in here. And this is done in close collaboration with Juan Aguilar and Juliana Martins from the National Hospital for Paraplegics, and are also an important part of our team in Piezo4Spine. But not only that, we also did an extensive histology of these animals. And again, we demonstrated this is also after 4 months, as you can see here, same time point as the previous results that I show you. We also get blood vessels. This is just the injury animal, and these are the 2 animals, the 2 groups of animals that have our scaffolds, those in which we found the neurons that were responding, and those in which we couldn't detect those neurons responding. And in both of them, as you can see here, the response was very similar and much better than in the case of the injury. So a higher and significantly higher number of blood vessels, and in case of this particular group, even the diameter. For the rest of parameters, they were similar to the injury. Not in the case of the neurites. We also evaluated neurites again inside our scaffold. As you can see here, we obtained a significantly higher number of neurites, length per neurite, and also total length of neurites when we were evaluated the lesion in which we implanted our reduced graphene oxide. And not only that, the distribution. Look at these diagrams. As you can see in here from the confocal images, we have a nice homogeneous distribution of the neurites all along the lesion side, which is very important because we want to replace, to regenerate the entire 3D volume, not only a portion, not only the lateral, the entire. Esther also added another parameter in the system that was motor rehabilitation. I mentioned before when I was talking about Ronaldo Ichiyama how important is motor rehabilitation. Why? Because it's a tool that we know helps the patient to improve. So let's add that in our animal models. That's what we did in here. We incorporated motor training, and what we found in all the cases and all the parameters, this is just a brief summary of all the results we got. These are under revision at the moment. What we obtained was either the preservation or the augmentation of the neural components and the reduction of inflammation. So we mitigate, we modulate a little bit the immune response. What is known from the clinical data that when we apply motor training, any physical activity, our immune system gets stronger and the response is better. We also incorporated 3D printing at this time, and that was the work of André Girau that came from the University of Aveiro to do some stages here with us. And he developed this very nice scaffold, very complex, with 3D printing, polycaprolactone, collagen, different elements, just trying to reproduce the structure of the spinal cord, where we get the gray matter in the center and the white matter around. That is the opposite what happens in the brain. In the brain we have the gray matter outside. Now that's the reason why the mechanical properties of the brain are different to those of the spinal cord. So by doing this mimetic system, we intended to have a better response in the spinal cord. He did a lot of in vitro experiments in which he proved that all the components of this sophisticated 3D assemble were biocompatible, all the different elements, the structure itself, and then also these sponges that he used with a different composition to resemble, to mimic the white matter and the gray matter. And the responses in vitro were fantastic. We moved to the in vivo and we did a comparison of the results with our foams and our microfibers. The response of the different components are here much better than the injury group. You see is this pink color, the different response for the different parameters that we evaluated, the markers, but it's still not better than the scaffold. The reduced graphene oxide that was simple, just a sponge, 3D porous, highly porous response, so we stopped the work in here. And then we move to another component that is also part of the Piezo4Spine project and is very important, that are iron oxide nanoparticles. We want to use them as nanocarriers with the capacity to respond to the magnetic stimulation as the delivery stimulus. We also have, as I mentioned before, the Italian Institute of Technology is developing very nice electrodes that want to apply that electrical stimulation to release the electrodes, to release the therapeutics. But we also have another component that will help us on the delivery, and it's been proved in our in vitro data that it can do that. There are these iron oxide nanoparticles. Of course, many of you already know and work with iron oxide nanoparticles, just to remind the fantastic nanosystems that can be for many different applications. They have been also tested in neural applications. You can see here how they have been used to alleviate Parkinsonian-like symptoms in mouse models. You see here the injection of the nanoparticles and then the application of an alternating magnetic field, and how those, from the behavioral point of view, get improved. Then we also have these systems for the successful delivery of stem cells. That is, stem cell therapy is being largely used in tissue engineering, and then also to combine the delivery of stem cells with the alleviation of symptoms by magnetic stimulation. Here comes Esther again with the piece of work that she has done with iron oxide nanoparticles, with the fantastic help, of course, of Puerto and Sabino. And in here, the particular focus of her work was to compare two different iron oxide nanoparticles, very similar or apparently similar in physicochemical properties, but with slight differences that we proved to have a substantial impact in the response of the neural cells that we had. You can see here both the NFA that we call, they have been synthesized in an autoclave, so a confined system where you can control more in detail all the parameters of the synthesis, and then the open traditional co-precipitation system that we call them NFT. So these, you can see from the pictures, are more multi-core-like, these nanoflowers, iron oxide nanoparticles, but in here, the more efficient heating made them to become more like a single core nanoparticles. In here we have the properties, and yes, there are differences, specifically more in the magnetic properties because of this sinterization, more efficient sinterization of the cores. Okay, let's move to test them with neural cells. That is our target. You see here, very nice viability is set for the ones that have been synthesized in the autoclave at this concentration, 0.1 milligrams of iron per milliliter. Is not that high of a concentration, but neurons are very sensitive. If you try these iron oxide nanoparticles with other cell types, you can probably reach higher concentrations, but not with neurons. They are super sensitive. And in here, we could see them very close to the cell membrane, to the neurites of the neurons, as you can see here, and also being internalized and inside our neurons. We also evaluated the process of internalization by using flow cytometry. It's a very useful and potent technique used in biology and biochemistry. What we found here, as you can see, basically the SSC is the side scatter that is telling us how complex is the particle that we are evaluating. So when the cell gets more complex, it gives you a higher value of the side scatter, and that's what we got. For those cells, you can see here, NFA and NFD treated cells, we could segregate the original population into two subpopulations. All of them increase the side scatter signal because they all get more complex because of the iron oxide nanoparticles attached to the cell membrane or internalized. But even that, we could still differentiate two. The ones that we call subset A, there was probably a higher amount of iron oxide nanoparticles interacting with these cells, and then the subset B. Because of this close interaction, we thought, okay, is the cell membrane being affected? Because we are really seeing a lot of nanoparticles inside, around the cell. So is this affecting the cell membrane? And indeed it was. As you can see here, we use this GP index, that is an index that tells you about the fluidity of the cell membrane of the in the different populations. We have here the subset A. Let me remind you that is that we think it has a higher dose of incorporated nanoparticles. In this case, we get, it's in here. The subset, it was mixed. If we look at the subset B first, that is the closer to the normal, the values got more negative. So already a significant difference. But if we go to the subset A that has a higher amount of nanoparticles incorporated or interacting with the cell membrane, it gets even more negative. So there is a clear impact of the iron oxide nanoparticles on the fluidity of the cell membrane. And that's important. Why? Because we are using this as nanocarriers to transport therapeutics. If we are changing the fluidity of the cell membrane, those therapeutics can enter more easily or more difficult. So it's important to take into consideration these specific actions that iron oxide nanoparticles have in the cell membrane, besides the action on other cell components. And now we get to Julia. And her fantastic work on iron oxide nanoparticles and magnetic hydrogels. She started this together with Marta Toldos, that was with us for 2 years and then joined the Universidad Autónoma. Julia developed this nice co-precipitation iron oxide nanoparticles and worked very hard to coat them with molecules that could be make them more biocompatible and nicer for delivery. Specifically, she selected chitosan and hyaluronic acid. There are two biopolymers with a lot of properties that are beneficial, and particularly beneficial in neural tissue engineering. So you have here the characterization and the magnetic properties that are not affected by the coating, and then the interaction with the cells. You see here again a very close interaction of these iron oxide nanoparticles with the different neural components, and again we could find them, both of them, inside the cells. We also check viability, the differentiation into neurons, into astrocytes, and there were not significant changes. And then incorporated, the next step was, as I mentioned before, we like 3D things because we want to replace a 3D element. So she incorporated that into collagen hydrogels. You see here how the color, collagen naturally is whitish because of the dispersion the fibers of collagen do of the light. But you see here how it turns more brownish because of the loading of the iron oxide nanoparticles. And indeed, in some details, you see here how we could find some groups of iron oxide nanoparticles in the borders. But naturally, they were well distributed in the matrix. We did transmission electron microscopy to prove that they were not just in groups in certain areas of the hydrogel, but homogeneously dispersed throughout the matrix. Again, we demonstrated the incorporation of those magnetic properties into the hydrogels because of the incorporation of the iron oxide nanoparticles. You see here the magnetization that was indeed dependent on the dose of the iron oxide nanoparticles of the loading. And then again, we culture neural cells on top of this, and we prove that the neurons differentiated into mature neurons without no troubles, and we had an important effect of the application of an alternating magnetic field to the differentiation of glial cells from this culture that we found a very interesting and promising data also for the in vivo implantation. And more recently, what she has done for the project is she tested also some of the coatings, as you can see here, not these biomolecules, but other, this is also a biomolecule, this APS. So different coatings just to make different options of coatings for loading and tested the biocompatibility. Unfortunately, these two were discarded, as you can see here, because of the bad neural response. So we confirm that these chitosan and hyaluronic acid were really the positive, the ones giving us the better response. And she loaded them with interleukin-4, that is a cytokine that is related to anti-inflammatory responses. That is something that we want to deliver in the spinal cord. We know that after the insult, the spinal cord gets rapidly a very strong inflammation, and then we want to control that and to drive that inflammation toward regeneration. So we thought interleukin-4 as a good target to be included. And she did not only test with neural cells, but also with inflammatory cells. We did with macrophages. Our colleague in Brussels did with microglia, and the results were very similar. And as you can see here, we are looking into, this is viability, so very high in all the cases in macrophages, not affected by the presence of the iron oxide nanoparticles. And then INOS, that is a specific marker of immune cells related to inflammation. As you can see, when we expose the cells to LPS, that is the lipopolysaccharide, is clearly a pro-inflammatory stimulus, is produced by bacteria, and our macrophages are trained by nature to respond to that, so rapidly increase the expression of INOS, the induced nitric oxide synthase, but also even in the presence of the interleukin-4 in the soluble form. So just adding interleukin-4 in the liquid, in the culture media, is still unable to revert the pro-inflammatory effect of the LPS. However, when we have any of our nanoparticles either loaded, functionalized with interleukin-4, or without the functionalization, we couldn't find that. And we proved that also by flow cytometry. As you can see here, the different profiles and then the numbers. So these are the numbers we get after LPS inflammation. We get mean fluorescence intensities around 8,000, but when we have our nanoparticles, we get down to 3,000-4,000. So a clear anti-inflammatory effect. We couldn't find that, just not to have everything positive, we couldn't find that with arginase 1. That is a marker of M2-like macrophages, so those macrophages that are more devoted to regeneration. On that, we couldn't find an impact. So we are still working and improving the system to really have something that can also impact on that side. And as you can see here, we could also find these iron oxide nanoparticles when loaded with interleukin-4 inside of macrophages, as you can see in both cases. So these functionalizations show a clear anti-inflammatory effect on macrophages, and also as demonstrated by our colleagues in microglia. So which are the conclusions for the progression of all these young people, very hard workers and motivated, that helped me to continue and progress in my career, also learning a lot from them. Ana Dominguez graduated in 2020 and is now at the second postdoc in France. Ancor González got a permanent position at the Hospital de Navarra in 2024 and is now a technician in there with his future, at least from the professional point of view, stabilized. André Girau is finalizing his first postdoc in the University of Aveiro and graduated in 2023. And Arche graduated also in 2023 in December and is now a technician at the Synchrotron Alba. Esther Benayes will be soon graduating, and Julia Martinez, we expect that also to happen after summer. Which are the take-home messages of these 10 years of a dream that is becoming a little bit of a reality, although the path is still very long, and we hope it's still long, to have also something to entertain, right? So from the scientific point of view, I hope I have convinced you, because I'm still convinced that graphene oxide and graphene-based materials in general keep promise for neural tissue engineering, and that iron oxide nanoparticles are very attractive nanocarriers for delivering therapeutics in the particular context of neural applications. From the professional point of view, what I have learned, that every grain counts, that it's important to take opportunities, but also to make them for others, not only to take advantage of them ourselves, and to believe in our capacities. When I first got the notice that we got the Pathfinder project and I was the coordinator, I was crying. I was crying because I was really afraid that I couldn't do. But then something inside told me, okay, you have been doing quite good in the past years, why not? Let's try, put your effort. Of course, it's not easy, it's a challenge. It's a lot of work, we know that. We have these type of challenges, but you have to believe in yourself and your capacities, because yes, you can, as is said. And from the personal point of view, it's important to save time for yourself, not to get exhausted. That is something that I fail often. To be generous with your mates. Yeah, okay, because we are all on board, and we all want to get our credit, right? Respect yourself and make the others to respect you, and bring empathy to work, because it helps a lot. And this is to be continued, I hope. These are very nice pieces of work that we have been doing all together, the Mambio group, that is a fantastic team. We get involved into many different activities, not only pure science, but also communication, so it's great being with them and really participating in all this. An acknowledgement, the whole Mambio team, the former members, those that joined us for some time, but did not wanted to stay for a PhD or something else, but they really learn and progress in their career. Elisa and the team in the National Hospital for Paraplegics, our current collaborators also to the past, and the funding agencies, including the Severo Ochoa, that we need to include in all our talks and posters and so on. And thank you very much to you all for listening and for coming to the seminar.
Science and how should be done. So my question is, you mentioned that you like the scaffold with the reduced graphene oxide because it promotes the growth of blood vessels and neurons. Mm-hmm. So the question is twofold. One, you have an explanation of why this scaffold works in a good manner, and if the performance of the scaffolds are better if done by using another hydrogels, collagen-based, or another biocompatible-based material.
Okay, thank you, Ricardo. Very nice question, because we keep wondering to some extent what is the response. For the first question you made, I think it's a combination. From my experience, also thinking about other materials that we have tested, is a combination of very nice mechanical properties, because despite of the great mechanical properties that graphene itself has, the truth is that in the shape of the scaffolds that we fabricate is a very soft structure, and that is close to the mechanical properties of the spinal cord. That's the first positive point. Then we also have a surface that has a lot of capacity to absorb proteins. So when you put that into the spinal cord, there are a lot of mediators that are released from the blood that get absorbed, and also help probably to make the cells to believe that the surface is nice, is biocompatible, and is something where you want to grow. That's probably also helping. It's not inducing, at least not to the extent of the time that we have explored, that is 4 months, any toxicity or any rapid reaction of death, affectation of the cells, what is a bio-inert surface, what is also very positive, and the fact of being porous and permitting the cells and the proteins to be deposited and the cells to get internalized in the 3D structure also helps a lot. It could be a nicer, more detailed explanation, probably. We haven't been able to really demonstrate what is in there that makes it different to others. From our experience, all the materials that we have tested, agarose, gelatin, graphene oxide in the shape of microfibers, many different variants, none of them has provided better responses than just the reduced graphene oxide by itself. We are now testing, in the project, this scaffold supplemented with the therapeutics that we are developing. So these iron oxide nanoparticles functionalized with interleukin-4 that I was explaining, together with some nanotherapeutics specific for the piezo-mechanoreceptors, all that combined in the same scaffold, and we expect the response to be better. Because if just the scaffold was good, just having extra stimulus should be better, but not with other materials.
Thank you very much for that excellent presentation. My name is Clint Achel. I'm the professor from Oklahoma State.
Nice to meet you.
And it's such a great opportunity for our students to see this presentation and to meet you. My question kind of touches on what you just said at the end there. Was there any attack or immune response from the body on the graphene? And did you see degradation of the graphene oxide over time, or did you do like cycling studies or long-term studies to see mechanical degradation? So it's kind of a two-part question.
Okay. Yes, thank you very much. Nice to meet you, and thank you for being here today. Hope you enjoyed. I was trying myself some part of my postdoc in the US, so I was close to your system at Northwestern University. I'm very happy to be there. Regarding your question, we did, regarding the degradation, yes, we did. In those 4 months implantation, we studied how the scaffold was. First, we measure by using atomic force microscopy in collaboration with Ricardo the mechanical properties of the scaffold after 4 months of implantation. What we found is that the mechanical properties were a little bit increased, and not because of the reduced graphene oxide, but because of the infiltration. It was completely populated by proteins that were deposited and cells, and we couldn't separate, maintaining the structure of the 3D component. So the mechanical properties don't go much farther than where they were, what is good. And then we also check the degradation by using different techniques, transmission electron microscopy, FTIR, NMR, and what we found is that the structure was dissembling. So the walls of our scaffolds were opening, and the nanosheets were being separated from the walls of the scaffold and being internalized by macrophages and other immune cells that were present there. So yes, the structure is at least disassembling and becoming shorter, smaller in pieces, and then being processed by the immune cells. Regarding the first question of the immune responses, in one of the publications we found and identified that the population of macrophages colonizing the scaffold was divided into M1-like, that is pro-inflammatory macrophages, but there were also M2-like macrophages. So those macrophages that were pro-regenerative were also present, attracted by our reduced graphene oxide. We are now expecting to have a higher impact because our scaffold will be loading with interleukin-4 functionalized iron oxide nanoparticles. So we really want to reduce that pro-inflammatory response by means of these nanocarriers. Thank you.
Well, thank you so much for this great talk. Always is really a pleasure to listen back neuroscience. So I have one question. Can you explain or elaborate a bit more which can be the origin of those differences in the neuronal repair in those graphenes that you mentioned, that there are some that they are regenerated, yes, and some areas no? You think that maybe can be a matter of diffusion of those inflammatory factors such IL-6, that is the opposite of IL-4 in terms, yes, of regeneration?
Yes. You mean in the work in which we did, yeah, the electrophysiological? Yeah. Yes. That's also an open question. We keep working on that. What we really believe or want to believe is that it's a matter of luck. because
Okay.
electrophysiological studies are very difficult. Basically, you introduce a very small electrode in the reticular formation and expect that the neuron that has to respond because it's connected to your caudal stimulation will be close by your electrode. But in there, you have millimeters in size, and your electrode is microns in dimensions. So what we really believe and are pushing to prove is that it's just a matter of longer time that you need to record, and you will finally find neurons responding in all the cases, because all the scaffolds are exactly the same, characterized with the same properties, fabricated in the same batch, implanted in sister, in this case brothers, because are male rats, brothers rats. So there are no other differences. You do the histology and they look, as you have seen there, basically the same. There's a little bit more of the diameter of blood vessels, but it's only a little bit. That cannot explain that you have neurons responded in the brain or have no. So it's probably just a matter of the complexity of the electrophysiological that lasts for hours, and sometimes at some point the rat just does not resist, die, and you get what you get. In those rats we were lucky, we got them, very clear signals. In the other rats we couldn't. What is very, very important is that from all the rats that we did just with the injured, we couldn't find ever a neuron that was responding. And in the case of the reduced graphene oxide, in 1/3 of the animals, we could find easily those neurons responding. So I think, and we believe, our colleagues, electrophysiologists, are the experts, right? You are also electrophysiologists, the complexity of those. They really believe that it's a matter of the time that you get to really do the recording.
Okay. Yeah, that could explain. No, that it's really honest from your side to show all the different spectrum, even though there is more amount of the ones that are regenerating. And another issue is, for the last part of the nanoparticles, are you planning to do the same experiment but with a pro-inflammatory cytokine to compare, such instead of IL-4 for the regeneration with IL-6, to be able to compare the pro-inflammatory scenario with all the players, no?
Yeah, it's a very good question. I don't think we can do during the project because of we only have 1 more year, and what we really want to do during this year that is left is to see what is happening with this complete therapy that we develop in the rats. So we don't have time to prove many other things. It's something to consider, especially in vitro studies that don't take so long to really prove if you go the other way around, if you activate. And also it's important to note that this, we are still very naive in what refers to the immune system. So it's not clear. No, this thing about M2 and M1 and all the broad spectrum that the macrophages have, we are still learning. So probably a good cocktail of cytokines can have a better impact than just a single cytokine selected and included in the nanocarrier. But yes, definitively something to that can be considered and explore in vitro. That is always easy.
Thank you so much.
Thank you, Conchi. Great talk, for sure. I have I know your project and all the science is trying to control the anti-inflammatory process. That's why you use the passage and so on. But the final the final idea is that the self-healing of the neurons. I mean, to control the peripheric the interfaces and so on, for not to be, because I think that all the inflammatory process and so on destroy any self-healing of the human body. So the idea is that the neurons communicate between them and self-healing themselves without any help, and just by controlling the interfaces. This is the idea.
Yeah. Yes, is much of basically that. There are many things about the dialogue between the cells that we still don't know. And the communication interplay between the immune system and the central nervous system is still very unknown. Actually, when we attend the meetings, the neuroscientists and the biomaterial science meetings, you see that even the own experts don't really know know each other in the sense of the immune system go on one side and the neural system go in other. But actually, our neural system is full of immune cells, and those are the first responding. So it's not only that neurons communicate between them and communicate with astrocytes, that are already well known, but the macrophages also communicate with the neurons and the astrocytes. And there is now a very important pathway of research that is how the astrocytes are also dual. So astrocytes, that are the glial component, the main glial component in the central nervous system, has also a broad spectrum of phenotypes. And that's responding to what they are receiving from neurons, but also to what they are receiving from the immune cells that are there. Macrophages that are extra based from the blood vessels because of inflammation, but also the microglia. The microglia, that is the resident immune cells in the central nervous system, rapidly respond. So it's a very difficult communication between all the cell types. In our case, we are not, these results that I presented with the reduced graphene oxide, we did not include anything in the scaffold. And the neurites were just colonizing the scaffold very nice. We did not add any growth factor there to tell the neurons, please come in. This is nice. They just found it by themselves. So imagine what we could get if we also add those components. They're just really attracting them farther in a more abundant way. So it's that's the key point, what you mentioned, the communication between them. But we are still naive and very limited in the knowledge that we have between, in that communication between the different cell types and what mediates the dialogue.
Thank you.
So thank you very much for the excellent presentation. A very basic question from my lack of knowledge on the topic. I was always under the impression that when you have a spinal injury, it was the growth of scar tissue that prevented the regeneration. But you hinted at the beginning, and I think also in the answer to the last question, that it is not just a mechanical impediment, right? So it is not really the presence of the scar tissue by itself that is preventing the regeneration of the neurons. Is that right?
It's a combination, because at the beginning, that scar is not as hard, as difficult, such a real mechanical barrier at the beginning as it is chronically. So it keeps building because nothing else stopped that. Actually, there are very nice work by Professor Friesen that demonstrates that if you completely inhibit the formation of the scar, there is no regeneration either. So you need some of the scar, but just the enough appropriate amount and in the right amount, combination. What is being demonstrated is a little bit of the scar and also the type of molecules that are forming the scar. So, as you mentioned, it's a combination. Biochemical mediators in there that help that to grow, the position of proteins, cell death, there is a lot because of the mechanical insult. And the release of those components of the cells also tell the others to die. So there is a loss of survival of the cells that are in the lesion area that cannot regenerate at all because they are dying. So it's a much more complex system than it seems. That's the reason why there is no cure. Because otherwise, there are so much smart people in the world, scientists working on this for many decades. If simple, it could have been already solved.
Thank you.
Question?
Okay, thank you.